Good afternoon, everyone. I'm Forrest Ray, reporter with GenomeWeb, and I'll be your moderator today. The title of today's webinar is Interrogating Tumor Structural Variation and Nongenic Dark Matter with Whole-Transcriptome Profiling, and is sponsored by Watchmaker Genomics. Our speaker is Brian Pining, Technical Director of Genomics at Providence St. Joseph Hospital Health. You may type in a question at any time during the webinar. You can do this through the Q&A panel, which appears on the right side of the webinar presentation. If you look to the bottom tray of your window, there are a series of widgets to enhance your webinar experience. With that, I'll turn it over to Brian.
All right, thanks, Forrest. Hi folks, I'm Brian Pining. I am Technical Director for Genomics at Providence Health. And I run a research laboratory at the Earl A. Childs Research Institute in the Providence Cancer Institute. And let's see. And so I'm going to be talking to you today about a couple different ways we've utilized whole transcriptome sequencing in the Cancer Institute here. And let's see here. There we go. Okay. And so here are my disclosures. And I just want to note that I don't have any financial relationship with the sponsor, Watchmaker, other than I'm a user of their products. Okay, so just a little bit of background. So again, I mentioned I do genomics within the scope of the Providence Health System. So if you don't know about us, Providence is the third largest nonprofit health system in the US. We treat on the order of about 3 million patients spanning 7 states. There's about 40,000 new cancer diagnoses per year within the system. And then our work comes in, we support the entire health system through a combined clinical and research genomics core based here out of Portland, Oregon. And so one of the unique aspects of operating within the scope of a big health system is there's some unique opportunities both to provide clinical genomics at scale, but also research opportunities in rather large patient populations where we see all sorts of unique things such as rare oncogenic drivers, rare tumor types, et cetera. And so I'm going to be talking about a number of different applications where our genomics core has supported the new development of different clinical and research opportunities. And so the way we operate the laboratory, so we perform CLIA-CAP sequencing primarily of tumors for cancer patients across the health system with the goal of, again, informing clinical decision support. So identifying any mutations that are targetable with precision therapies, biomarkers for immunotherapy, et cetera. But at the same time, we also operate a research laboratory. And so a lot of our work is taking these clinical data, so clinical sequencing, imaging modalities, real-world data, derived from big databases, and working with that in a de-identified fashion, but also combining that with really non-clinical, more kind of novel omics assays. And so that spans everything from whole genome sequencing, RNA-seq, to structural characterization, ATAC-seq, epigenomics, et cetera, as well as really kind of the way the field's moved is into spatial and single cell applications. And then we see that this is a feedback loop where these developments in the research realm can then come back into the clinical lab and get deployed as novel assays. So that's a lot of the way we operate here. A couple novel things about our cancer genomics program. So a number of years ago, we introduced a reflex model, which is somewhat unique, but becoming more commonplace, where cancer genomics is initiated really at the time of diagnosis for advanced cancer. And that is initiated by the pathologist. And the goal really was to make access to the genomic profile as early as possible in clinical decision making. So trying to get the genomic results in the hands of the oncologist really at the first meeting with the patient, so that could inform decision making from the get-go. And so kind of what that looks like. So, traditional workflows, patient has a biopsy, it's evaluated by pathology, there's a diagnosis that's made. There's some treatment planning with the oncologist. The oncologist will order testing, comprehensive genomic profiling, and that's performed and then returned. So the thing we're trying to remedy is making those results available earlier in that decision-making timeline. And so we've come up with this pathologist-directed workflow, which has this done routinely at the time of diagnosis by the pathologist. So we're trying to get the results in the hands of the treating physician right away. And so the assay we use in the clinic is an assay we call ProvSeq. It's derived from Illumina's TSO 500. It's a DNA RNA hybrid assay, a targeted assay. And the goal is, again, really to assess for mutations across a spectrum of a large panel of cancer genes. And so, the different tumor types we look at kind of really mirrors the patient population and is similar to, what you would see across cancer centers throughout the world. And so just looking at some of the alterations we observe in the course of routine clinical practice, really this is kind of looking across tumor types with the pointer up here. Yeah, but the magnitude of the circle indicates, the percent of cases that test positive for these biomarkers. And so, common biomarkers, PD-L1 high, TMB high, and then, usual suspects, BRAF and melanoma, et cetera. The one thing I do want to point out is a large number of these oncogenic drivers are actually fusions, so fusion genes between a proto-oncogene and another fusion partner that drives the expression of that gene. And so, these include ALK, NTRK, ROS1, RET, and many of these are highly actionable. And so, again, the unique challenges around identifying oncogenic fusions is they're occurring via the fusion of two different genomic loci. More challenging to detect via short-read NGS than traditional SNPs and small indels. And again, so this is kind of looking at the spectrum of fusions we see. This is a Circos plot across all chromosomes in the human genome. And then they're color-coded by inter and intra gene fusion pairs. And so a couple, you can see a couple highlighted genes such as NTRK, we observe fusions with a variety of fusion partners. And so because of these challenges, RNA-seq has really emerged as the preferred method for detecting these fusions. And so, and a lot of that derives from things that, what I just said, but also, fusion breakpoints can be highly variable. And so using a DNA sequencing approach, you really have to deeply sequence really long contiguous stretches of DNA just to make sure that you don't miss a key fusion breakpoint. Whereas RNA-seq, right, because you're looking at the mature RNA product of that fusion, gives you kind of the unique advantage to sequence across that breakpoint region. And that's really what is important for us for, number one, mapping where that breakpoint occurs, but also whether it renders the oncogene in frame as well, and gives you some sense of expression level for that fusion too, because the level of RNA, is relevant to the amount that gene is being expressed by those cells, unlike really the DNA content. And so, like I mentioned, some of these fusions are highly treatable with precision drugs. This is just data from an older paper, one of the initial studies of larotrectinib. So it's an anti-TRK inhibitor, so TRK fusions, where you see remarkable durable survival in patients that respond to this drug. And so a large number of patients harboring these fusions respond, and that response is highly durable, which is really kind of remarkable in terms of the spectrum of cancer treatments. And indeed, like in our practice, we've seen really kind of remarkable events where patients present with metastatic disease and are on hospice, and we find a TRK fusion, and the patient responds dramatically and is taken off hospice and goes back to living their daily life. So really kind of remarkable stories you can see by detecting these types of alterations. And then overall, the use of large-scale genomic profiling, comprehensive genomic profiling with RNA-seq, at least in our practice, we've seen a pretty dramatic improvement in survival. So this is looking at population advanced lung cancer patients, comparing patients tested with comprehensive genomic profiling versus prior small gene panels or single gene testing, and likely due to the additional targets that you can detect, there's more treatment options available to those patients. Okay, and so within the scope of our patient population, we've observed a variety of fusions over the last few years. This is just looking at the scope of NTRK fusions we've observed. So, so NTRK 1, 2, and 3, as well as, looking at the different varieties of fusion partners we've observed to date. Relatively common fusion partners such as EML4, but also, indicated with asterisks are are fusion partners that are novel, right? So if, depending on the type of assay you're using, you may not be able to detect those fusions because you may not have a probe targeting that fusion partner. And then again, so combining DNA and RNA makes a lot of sense because, each cancer case has a variety of different alterations. Some of them may have multiple targetable alterations. This is just looking at other alterations in common with NTRK fusions. One of the things we observed in our data set is a number of these cases are highly mutated, and so also have high tumor mutational burden, and so potentially could also be targetable with immunotherapy. Okay, so one of the things we've looked at is adding, again, we use a targeted panel currently, but based on how the field's evolved, we're currently looking at adding whole transcriptome sequencing to the assay, and so for a couple different reasons. Let's see here. Okay, so anyway, versus targeted sequencing, whole transcriptome sequencing gives us, obviously, you can then use that to develop gene expression classifiers, and we've seen a number of these, gain relevance in clinical practice, and more are currently being characterized or studied in trials. And so adding whole transcriptome profiling allows you to do that. Also, you can look at, the effect of splice donor acceptor mutations on the downstream transcript, et cetera. And so again, it gives you a lot more additional data that could be useful in the clinic, but would definitely be useful for downstream research studies. And in a system as large as ours, having whole transcriptome data on a wide variety of patients could potentially lead to novel discoveries. The big challenge with that is the way clinical pathology is currently practiced around the world. The vast majority of tissues we sequence are FFPE, so formalin-fixed, paraffin-embedded tissues. And so those, formalin can have dramatic deleterious effects on nucleic acids in terms of inducing damage. And so that can be a challenge for a variety of assays, DNA and RNA assays. And so a couple of the main methodologies for RNA-seq include poly-A selection. So again, that's typically like oligo-DT priming. And the goal is really to enrich for non-ribosomal RNA products. So ribosomal RNA makes up around 90% of your RNA pool, and so the goal of really of these enrichment strategies is to kind of get around that RNA and focus on messenger RNA. Other assays include exome scale capture. So this is using a exome scale probe set to hybrid capture all genes of interest, and that works quite well. As well as ribodepletion and whole transcriptome sequencing. So this is typically using beads to capture the RNA pools out of your sample and then removing them. And so how these apply in the scope of FFPE tissues. So again, poly-A selection based on the nature of that approach is generally incompatible with FFPE due to the fragmentation that we see in these transcripts. And then we're talking quite low RIN scores from FFPE-derived tissues. Exome capture, it works great. Typically there's a variety of different kits for doing this type of capture. It usually works quite well for FFPE, but the challenge there is it does not include novel transcripts, so non-coding RNA, et cetera. And what I'll get into shortly, the way our research has evolved has really been looking at some of this, non-coding RNA. And then ribodepletion and whole transcriptome sequencing, there can be challenges there as well. We've had kind of mixed success with a variety of kits from different manufacturers. And so overall, like what we're looking for is going towards a single whole transcriptome assay that we can use for multiple applications. And so it had to be compatible with FFPE, really had to have the ability to sequence small biopsies, so less than 10 nanograms in input. And again, our focus was to support the use of whole transcriptome sequencing for the detection of novel transcripts. And so moving away from probe-based approaches. And then again, a rapid workflow to enable throughput. So we've evaluated a variety of different kits over the past couple of years. And one that's really worked for us in our hands in the lab has been using the Watchmaker RNA-seq kit. And that includes a couple novel steps. So there's a ribodepletion step. So using this Polaris mix. So it's a ribodepletion plus globin depletion step. Real straightforward. It's essentially a relatively short hybridization time and then a digestion and bead cleanup. And then the downstream protocol from that, again, is very standard, in the field in terms of RNA library prep. So again, RNA globin depletion, bead cleanup, FFP treatment, fragin prime, first strand synthesis, second strand synthesis, ligation, bead cleanup, AMP, bead cleanup, and then quant normalized pool. So very, very, straightforward workflow. I think the unique thing for us is if you look at these times associated with these steps, it's really quick. So we can generate libraries in a very short period of time, which is very advantageous for a high throughput lab such as ours. And so what do the data look like? So we evaluated a variety of clinical FFPE samples that had previously been characterized to harbor pathogenic fusions, and just showing a couple examples from this. So on the top, this is the Watchmaker total RNA approach versus our standard hybrid capture ProvSeq assay. And so this is for those of you not used to looking at this is an IGV window. We're looking at read pile up, so all the reads are mapped to the specific exons in NTRK1 across chromosome 1 here. And so you can see that the two assays look, there's slight differences, but they look relatively similar. If you look at the histograms for the read pileups, they look fairly similar between the two different assays, one being a capture and one being a total RNA-seq assay. And so going to fusion detection, and so we generally use star fusion for fusion detection as well as MANTA. If you compare, if you zoom in on fusion breakpoint, so this is exon 11 in NTRK1, and what we're looking at here, the kind of the rainbow color, these are mismatch bases. So we're looking at essentially bases that properly map to NTRK1 on the right. And then at the end of the read, you see these mismatch bases. And essentially, if you take this sequence and blast it, it'll map to the fusion partner, lamin A. And you can see that there's a large number of supporting reads in both assays. And these are the types of reads we're really looking for because they span the junction breakpoint. So it allows us to map the actual fusion breakpoint and characterize whether that fusion's in frame or not. And then similarly, looking at the cognate, so this is lamin A, this is exon 10, and you see for Watchmaker, again, here's this these mismatch bases that map to NTRK1, and they're seen kind of in both assays. Looking at another example, the ZTV6 NTRK3 fusion. Similarly, we see, this is zooming in on the NTRK3 locus, I think this is exon 15 on chromosome 15. And you can see, again, you see these mismatch bases. And this is kind of how you can quickly view these manually in IGV. There might be better visualization tools. Feel free to put examples in the Q&A if you have a better tool for visualizing fusions. But this is one of the ways we do it in the lab here. And then kind of zooming out, you can kind of see the differences between the capture-based assay and the Whole-Transcriptome assay. There's kind of different uniformity in the reads based on whether they're derived from a specific capture probe or not. And then, again, while they look relatively similar, there's also, kind of additional content that kind of lies outside of the capture window. In this case, it's relatively low level. And then looking at the fusion partner ETV6, again, we have these reads that line up really kind of in both assays, and again, mapped to the NTRK3 gene. Okay, one additional example. This is a SDC4 ROS1 fusion. This is looking at exon 6 of ROS1. And again, similarly, we see these reads mapping in both assays. Kind of zooming out, one of the things that's unique, here on the bottom is the capture assay, but in the whole transcriptome assay, there's a lot of additional transcription that seems to be going on. If that's and that's either pre-splice mRNA or other types of non-canonical expression. And then again, looking at the SDC4 assay, similarly, you can detect junction spanning reads from both assays. And then kind of zooming out, you can start to really see the difference between a capture and non-capture assay. So SDC4 is detected, in the capture assay, it's detected based on capture from the ROS1 probe. But in whole transcriptome, you're not probe based, so you really have, the rest of that gene reads mapping to the entire gene, and of course that's again translates across the whole transcriptome as well. We have the additional content of the transcriptome backbone. So just from this first part, just a couple quick conclusions. So we can use we're able to successfully use whole transcriptome sequencing to characterize clinically relevant fusions in FFPE samples. And so the assay potentially makes a nice partner to a targeted assay where it gives you that whole transcriptome backbone that could potentially open up new clinical and research applications. And again, some of those applications that have emerged include gene expression classifiers, detection of novel transcripts, and then kind of building cohorts for characterization of tumor-specific differences across patient populations in terms of treatment response and other applications. Okay, so switching gears, I'm going to talk a little bit about a novel research application using whole transcriptome sequencing in our group. And it's really in the context of immunotherapy and in terms of characterizing what we're now calling cancer's dark matter. Or this concept of non-canonical peptides that are potentially immunoreactive and could be targeted by the immune system to kill cancer cells. And so, what are non-canonical peptides? So non-canonical peptides are derived from, essentially non-canonical genes. And so they can either be, the traditional neoantigens are derived from tumor mutations that then get presented by the immune system. But there's all this additional stuff that gets expressed, we think aberrantly, by tumor cells and then gets translated. And those can actually get presented by HLA1 as well. And so a lot of it's really short-lived and can get rapidly degraded. But what you can do is you can actually develop a vaccine. You can potentially develop a vaccine where you can immunize against these things as a way to target the immune system to that individual's cancer. And so I participate in a study where we're utilizing a couple different approaches in common to really enhance tumor-reactive immune responses. And so that includes, again, using a cancer vaccine plus a T cell agonist. This is anti-GITR in this approach, as well as, essentially removing the brake. So removing the immune checkpoint activity via anti-PD-1 therapy. And so working with my colleague, Bernie Fox, where his lab developed this dark matter vaccine. And so, as I mentioned, these kind of short-lived proteins or peptides get degraded rather rapidly, but you can block that with essentially proteasome blocking drugs. And so you can lead to the generation of these long-lived products that you can then package into a vaccine. And so that's exactly what Dr. Fox's group has done, again, in collaboration with the company UbiVac. And let's see here, there we go. And I'm not going to go into too much depth in terms of the composition of the vaccine, but it was recently launched into an immunotherapy trial, which essentially includes kind of the three things we talked about. So vaccine, anti-GITR, and anti-PD-1. And so this was a trial that was developed and launched by, again, so Dr. Fox, as well as Dr. Marcus Cooey and Dr. Ram Leidner here at the Providence Cancer Institute. It's underway. And, again, so there's multiple treatment arms. There's a variety of immune readouts where kind of my group comes into play is, again, doing RNA-seq on these samples as well. And so initial results from the vaccine or from this trial look, positive. This is data that was presented at AACR earlier this year, where this is looking at one specific HPV positive patient. And essentially, here's an example where you can see significant shrinking of a lesion. So again, the study's rather early, but initial results look promising. Where we come into play, much of our work is focused on trying to understand which dark matter non-canonical antigens the immune system is responding to. And so again, the vaccine is comprised of cell lines made by, along with enriching for these dark matter transcripts, peptides, et cetera. And our work is using whole transcriptome sequencing to characterize these things. And so, what are we talking about? So these variants can arrive from a variety of different things: exon skipping, intron retention, non-coding RNA expression, defective translation, et cetera. So there's a variety of different ways these things can arise. And so what we developed was a pipeline for characterization of these things, both in the vaccine itself and then downstream, kind of in patients as well. And so how that works is we perform total RNA-seq to generate essentially a library backbone for both coding, standard coding transcripts, but also these new kind of open reading frames as well. And so that includes standard kind of assembly, variant calling. We do a multi-frame translation, and all that leads to a patient-specific database. And really the end goal for that database is to be utilized in combination with mass spec, so actual detection of these peptides. And so one of the significant challenges with immunopeptidomics is the sheer number of possibilities for standard for non-canonical peptides, and it really creates a peptide searching database that is is essentially huge, and it leads to it can lead to high false positive rates. So the strategy we've come up with and other folks in the field have come up with is utilizing Whole-Transcriptome RNA-seq to create a patient-specific database where you really know which non-canonical targets are being expressed. And so that way, you're really shrinking the database search size for your mass spec-based kind of peptide searching, and leads to more positive hits. And so we've done this across a number of different patient-derived cell lines. And so looking at the number of unique peptides we've identified from the mass spec data mapping to these RNA-seq derived databases, broken down by HLA supertype. And so, the take home is, the majority of peptides are coding, right? They're standard coding peptides, but you have this, this stuff at the end here, which is derived from a variety of different things. And on the right side, if you zoom in, you can kind of see the different breakdowns of these different constituents in terms of, the non-canonical peptides. And so some of them are derived from 5' UTR, 3' UTR. We also have non-coding RNAs, a much lower frequency of intergenic. We get intronic or off-frame detection as well. And then characterizing, these non-canonical peptides across different patient-derived cell lines. We can see, looking over here, you can see that, this is looking at all peptides. There's a high degree of commonality amongst across the two different patient-derived cell lines, but a lot of unique peptides as well. And then, and again, like within looking at non-canonical peptides, it's a small majority or a small minority of peptides in the total pool, but also of note, a number of these are shared across patients, which is interesting. And then looking at, across 3 different patient cell lines, like this is looking at non-canonical peptides that are shared across multiple patients. And, you can see there's, depending on HLA supertype, there's a number of these that are shared across multiple patients, which is potentially interesting. So that work is ongoing, but you can kind of see where we're really kind of plugging in whole transcriptome sequencing as a way to really kind of unlock the potential of downstream kind of mass spec-based immunopeptidomics to really find these things. And kind of the ongoing work in the lab is doing kind of more in vitro assays to discover, which of these are truly immunogenic. But from kind of early clinical data, this looks like a promising approach. So a dark matter-based vaccine does appear to lead to tumor shrinkage, and at least in early trial results, that's still ongoing. But I think where we're going is towards kind of models where you have patient-specific vaccines kind of derived from derived from characterization of that patient's unique tumor genomic profile, where you can develop really kind of personalized treatment plans for enhancing immune responses in combination with immunotherapeutics. And then with that, I'd like to thank a variety of folks in my group, as well as other groups. A lot of the RNA work was done by multiple bioinformaticians in my lab, Alexa Dowdell and Ryan Meng, as well as in the core. I'd like to highlight Ben Cosgrove, who helped us with a lot of the pipeline work, as well as in the wet lab. A variety of folks helped, but I want to give a shout out to John Welly, who did most of the RNA-seq experiments, as well as my key collaborators in the Cancer Institute, Bernie Fox, Ram Leiter, Marcus Cooey, Yoshi Kaguchi, as well as our collaborators in terms of mass spec data, both with folks at Shimadzu Scientific and the Japanese Foundation for Cancer Research, and also my colleagues at Watchmaker in helping us get up and going with these assays in the lab. And I can take questions as well.
Great, thank you, Brian. As a reminder to webinar participants, if you have a question, please type it into the Q&A box in the control panel. We'd like to ask attendees to take a moment after the webinar has ended to take our exit survey and to give us your feedback. We will now start the Q&A portion of the webinar. The first question is, what does FFPE treatment do? Is it an enzymatic reaction? And why is fragmentation necessary for FFPE in the workflow?
And I can take that one. I'm a product manager for Watchmaker, so I can definitely give some details around the chemistry itself. The FFPE treatment step is really there to help facilitate additional sample cross-linking if there is any present. And the root of it is that all extraction methodologies aren't built the same in terms of their efficiency and ability to reverse residual cross-links. And so this treatment step just essentially helps to level the playing field across different extraction approaches. So it's good for labs that are taking in maybe pre-extracted samples so they don't control that extraction chemistry part of the workflow, or folks that are working with maybe just an inefficient extraction solution. So, yeah.
Yeah, and I'll add to that. In terms of the frag step, FFPE fragments nucleic acids non-uniformly. So the frag step's really getting, molecules that are suitable for essentially generation of a sequencing library.
Great, thank you. Our next question is, what type of methodologies do you recommend, with or without single-cell sequencing, that allow for comparing and complementing the expression of mRNA and non-coding RNAs from the same sample in order to better reconstruct the co-expression networks between them starting from biopsy samples?
So I'd say strategies for bulk sequencing versus single cell sequencing are potentially quite different. Commenting for bulk sequencing, again, like if in general, you're really only going to see non-coding RNAs if you're doing whole transcriptome sequencing, unless you've developed, a probe-based panel that's specifically that has probes that are going to hybridize to those non-coding RNA. One of the things we've seen in our data, is these RNA-seq libraries are, very robust in terms of minimal ribosomal content, very diverse, detecting large number of genes. And the quantification of those genes across replicates is highly reproducible. So we've used it in a variety of studies in terms of, kind of exactly what you're asking, kind of reconstructing co-expression networks. And so using these as, for example, input to WGCNA-weighted gene co-expression analysis. And you can really generate, clusters that have functional relevance based on the data, which is what we're expecting to see in these types of data sets.
Great, thank you. Our next question is, what is your experience with fusion transcripts that result in decreased expression levels?
Yeah, so a fusion transcript that is resulting in a decreased expression level is interesting and maybe less clinically relevant. Often what we'll see is we'll see the fusion transcript results in a much higher expression level. And so if the level's decreased, we may want to confirm via IHC or FISH or some other type of assay to see if it's producing a functional fusion protein.
Great, thank you. Our next question is, do you see an advantage to targeting dark matter over neoantigens that come from canonical sequences, I'm guessing?
Yeah, so, I mean, they're different strategies. And, so one of the challenges, again, of developing essentially vaccines that target canonical targets such as mutated neoepitopes is there may only be a relatively smaller number of those within a patient's tumor, and those canonical neoepitopes may not be conserved across multiple patients. So one of the questions we're asking is, well, are there commonalities in the expression of non-canonical epitopes across patients? And I think the field is still relatively new in terms of deeply characterizing these across a variety of tumors and across a variety of patients. But there is some evidence that we're starting to see that these are shared, and then those that are shared are potentially immunogenic.
Great, thank you. Our next attendee asks whether the library kit can detect smaller RNAs like microRNA.
We usually use a different strategy to detect microRNA. I don't know if Jen has any additional comments.
I was going to say the same thing. We wouldn't recommend this workflow for microRNA detection.
Great. Kind of similarly, the next question asks, will this method detect highly structured RNAs and very long RNAs?
I will say I don't have, oh, go ahead, Brian.
No, go. I'd say potentially yes. I mean, our application is really an FFPE where everything's, highly, relatively fragmented and denatured. But I don't see a reason why, why we wouldn't necessarily detect very long RNAs. Our downstream sequencing strategy is using short read sequencing. You might want to introduce a long read sequencing strategy to try and generate contiguous reads across a full long non-coding transcript.
Great, appreciate it. Our next attendee asks, did you manage to obtain the TCR or IgG repertoire from the FFPE RNA-seq data?
Yes, I mean, you can get limited content from bulk RNA data. We will typically pair Whole-Transcriptome RNA-seq with a strategy typically targeting repertoires as a way to enrich for that. But you can see some limited transcriptional content in the Whole-Transcriptome library. So it kind of depends on your application.
Thanks. The next question is, what are the major differences between whole genome sequencing and exome sequencing?
Yeah, so whole genome sequencing, depending on the strategy, right, is going to give you a rather kind of unbiased look at the nucleic acids in your pool. You're not doing any significant selection upstream. And so, you know, versus exome sequencing, right, where you have some pool of probes that you're using in a hybrid capture strategy to enrich for specific regions of the genome. Exome sequencing, you know, obviously you can do for much reduced cost versus, you know, whole genome sequencing, which is especially important in tumor sequencing strategies where we're sequencing rather deep, so 400x, 1000x, et cetera. But there's definitely some drawbacks, right? So you're not characterizing any tumor alterations that don't occur in exons for the most part. The ability to detect structural variants can be somewhat more limited. And then, as we mentioned, you know, some of these fusion breakpoints occur in intronic sequence. And so you may have challenges in potentially mapping those.
Cool, thanks. Our next question is, what are the major factors that affect the quality of FFPE for DNA or RNA analysis?
Yeah, so the major factor, there's a variety of factors that can affect FFPE quality. Time and formalin is one of them. So you can overfix specimens, which will lead to additional degradation. Age of the block may play a role. Age of the slide, like if you're, or age of the slide scroll, you know, essentially the piece you've essentially kind of sliced off that block, you know, can get exposed to, you know, to air and lead to additional issues. One of the issues that really causes problems in the lab is decalcification reagents. So tumor fragments that are mixed in with bone. You know, additional enzymes are used to decal those samples, and that can lead to additional low quality of the downstream nucleic acids.
Cool, thank you. Our next question asks, what are the biggest obstacles to deploying whole transcriptome sequencing clinically? Yeah.
So you know, the biggest obstacle there's I'd say from a workflow strategy, it's, you know, I don't think it's a big challenge to deploy whole transcriptome sequencing alongside, you know, traditional DNA sequencing or, you know, targeted RNA sequencing. If you've already worked out strategies to extract nucleic acids from FFPE and can make libraries, you know, with kind of these modern enzymes and reagents for whole transcriptome sequencing in FFPE, it's relatively easy from a bench-based perspective. Informatics, you know, there's additional pipelines that need to be built in terms of fusion detection and then any gene expression characterization. And then there's the factor of cost. So if you're, you know, utilizing whole transcriptome sequencing for, you know, routine work in the clinic, you know, such as fusion detection, you know, developing that into a panel that, you know, is, you know, essentially approved by payers, et cetera, and reimbursable. You know, I think those kind of factors need to get get planned out ahead of time.
Great, appreciate that. Our next attendee asks, do you perform macro dissection to enrich for tumor fraction or scrape the whole slide for lysate preparation?
Yeah, we always perform macro dissection. And so the reason for that, and, you know, for those of you not doing this on a routine basis, so your tumor biopsy, your tumor resection specimen likely includes some combination of tumor cells and then kind of and then the surrounding normal tissue. And so that surrounding normal tissue, any significant amount of that can dilute the presence of any variant, be that, an SNV or indel or fusion transcript, because you have this pool of essentially normal cell-derived DNA or RNA. And so what we do is macro dissection. So every specimen a pathologist reviews, identifies regions rich in tumor, and then one of us in the lab will go in and actually use a scalpel to remove specifically that region from the slide. Additional strategies are laser capture microdissection, which can get you an even more pure tumor pool, but also introduces additional workflow time.
Appreciate it. Next person asks, what is it about the Watchmaker kit that makes it appropriate for FFPE whole transcriptome sequencing?
Yeah, Jen, do you want to take that?
Sure. I would say during the development of the workflow, a lot of time was spent focusing on performance with FFPE. And so I think there are a couple of novel features that I think boost performance. One is what we already talked about, which is that FFPE treatment that's there kind of as a safeguard for variable performing extraction methods to really, again, facilitate additional cross-linking reversal. The other is that we actually undertook a reverse transcriptase development and enzyme engineering project in parallel with this RNA-seq solution. So we were feeding a variety of novel variants in for RNA-seq screening. And we really selected one that overall performs really well in RNA-seq applications. And it boosts the conversion of your RNA to cDNA. So it's, again, just giving you, better performance. And then the last thing I would say is that we spent a lot of time kind of fine-tuning the workflow itself, and these things aren't just specific to FFPE. It's performance with low inputs or challenging samples in general. But this idea of minimizing the number of purification steps and handling steps in the protocol, it minimizes user error, but it also minimizes points of loss in your system. So you can lose material non-specifically at every tube transfer step and at every cleanup step. And so if you can really reduce those, it makes it for an easier protocol, but it also, again, can improve the actual data quality at the end of the day. So I think all of these things really come together to make it a suitable solution for FFPE.
Yeah, and I'll just say as an end user, we've found in our hands that we've used the kit to make really kind of diverse, robust libraries from low input samples that we didn't have success with other assays.
Great, thanks both of you. And that's all the time we have for today. I'd like to thank Brian Pining and Jen Pavlica and our sponsor Watchmaker Genomics. If we didn't have time to get to your question, we will try to follow up with our experts. As a reminder, please look out for the survey after you log out to provide your feedback. If you missed any part of this webinar or would like to listen to it again, an archived version will be mailed to all attendees. Thank you for joining us for this Genome webinar.